And we left last class, we were on this slide, classifying epithelium. The general arrangement and type of cells dictate what type of epithelium you're dealing with or what type you're looking at. And so the arrangement of layers can either be simple, there's one layer. It can be pseudo-stratified, which is, it's still simple. There's only one layer of cells, but they're shaped irregularly. It looks like they're stacked, or it looks like there are multiple layers, but they're not. There's one layer. Some of them don't go all the way to the surface, but we call that pseudo-stratified. And stratified is multiple definitive layers. Easy to determine or identify, multiple layers of cells. Now, the types of cells also influence the type of epithelium you're looking at. Squamous is very thin, and it's the type of epithelium you typically find in the lungs, where you want a thin membrane or thin surface to allow the movement of gas, oxygen and CO2 in that instance. So you often have squamous epithelium there as well as in other places. Cuboidal is so named because the cells look like cubes. They're square. The larger the cell, the more space there is inside for things, for enzymes, for organelles, for storage and processing. So the kidneys typically have, and many glands have cuboidal cells in order to produce various things that need to be secreted or process things as they move through the tissue. And columnar are long stretched out columns, more space to store and process items. And the digestive tract often has cells that look like that. Now these can come in many different arrangements, many different combinations rather. You can have stratified squamous or simple squamous epithelium. You can mix and match these characteristics as you see fit. The name, though, depends. Sometimes you have multiple different arrangements in an epithelial tissue. The name depends on the most superficial layer. The most superficial layer. The specific type of tissue, if it's multiple layered and stratified, depends on the most apical, or the cells that are closest to the surface, the most superficial layer. And these figure references are updated for your book. Some of the references in the slides online haven't been updated, but these ones have. 4.6 and 4.8 in your textbook have a great summary of how to name various tissues based on those cells. So this one, if you're paying attention, is stratified cuboidal epithelium. Right? The cells look like small cubes, maybe they're rounded. There's definite layers, 4 or 5 layers. Stratified cuboidal epithelium. Pretty straightforward. This next example is not as straightforward. This is an image not unlike what you'll see in lab next week. And the epithelium in this picture is the layer at the top here. There's connective tissue below that. But we would be tasked with identifying this epithelium. This might be something that you see on the lab midterm exam, very likely. Maybe not this exact question, but something like this. This isn't the best picture. The resolution isn't as high as we would like, but let's work backwards. Knowing this is a more complex type of epithelium, ciliated, pseudostratified columnar epithelium. Let's see if we can see if that makes sense. At least one part of that won't make any sense right now, and that's fine. But let's take a look at the epithelium and see if we can make it make sense. It looks fairly thick. The band looks pretty thick, so automatically I'm thinking it's either stratified cuboidal, because it's thick, or it's columnar, because those cells are long. The giveaway that differentiates between stratified versus not is the position of the nuclei. In stratified epithelium, you're going to have many distinct layers of nuclei, little dark staining dots, and they'll all seem ordered. We don't see that here. And I'm pointing with these red arrows to different nuclei that seem to be at multiple levels. They're not in ordered rows. And so that tells us these are pseudostratified cells. Some cells span the entire width of the epithelium, some get stuck earlier. The nuclei are in different regions. There's not articulated layers. So this is pseudostratified, and hence because of the thickness of these cells, they're columnar. They're obviously not cuboidal. The ciliated portion comes into play. If you could zoom in on this upper layer, it looks really fuzzy. And cilia, we'll return to in the digestive system, are small finger-like projections on the ends of the cells. And they're really good at increasing surface area so that fats, carbohydrates, proteins can be absorbed. Little finger-like projections that here just look like fuzz. And if you saw a higher definition picture, it would be a bit more obvious. But that layer of fuzz on the apical side indicates that these are ciliated. So working backwards, this makes sense now. And you are going to work forwards in lab and during your exams to be able to name these appropriately. What will help you a little bit is this cheat sheet table that I put together. These are the combinations that are possible. These should be in your notes. Notice you can't have pseudostratified squamous epithelium. The X's are all the combinations that are possible, and what we haven't talked about yet are transitional cells. We don't often focus on those because transitional cells can change shape. Think of your bladder. If you've been holding it in for a long time, your bladder can distend and stretch, and the cells of your bladder can actually change shape. They might go from cuboidal to columnar. They're transitional. It's really hard to tell if a cell is transitional from a microscope section, from a microscope slide, from a 2D image frozen in time. You can't tell if they're transitional. So we don't often look at those in lab or during the exams. But the others you should be able to name no problem. So we're pretty comfortable with epithelium at this point. The lining epithelium. The upper surface, facing away from the body or into a cavity, the most superficial surface. But it's not only flat. Epithelium is not only flat. It's the surface lining of many tissues, but some structures are created by epithelium, known as glands. And glands are typically places where something is produced. It could be mucus to lubricate, it could be enzymes to break down, it could be sweat. All of those substances are produced by glandular epithelium. Glandular epithelium, cells that are producing a substance at any given point in time. It could be a single cell, or it could be multiple cells in a pit as shown here. Now, we won't go into too much detail about this, but just be aware that glandular epithelium, when we get to specific examples, we'll see sweat glands later today, and we'll see this a lot in the digestive system next semester. But glandular epithelium is broadly broken down into two categories, endocrine and exocrine. Endocrine glands deliver something inside the body, endo, into the interstitial fluid, that is the fluid between cells, interstitial, between cells. We can secrete substances into that fluid, and that goes into the bloodstream to be distributed throughout the body. Hormones, for instance, we'll see are secreted in that way when we get to the end of this semester. Exocrine glands, on the other hand, don't secrete into the fluid within the body, they secrete outside of the body, onto the surface of the body. And the best example there is a sweat gland, which you probably all have experience with. A substance is secreted on top of the skin surface. It's outside of the body. Sweat glands are exocrine glands. Also of note, glands in the stomach. They're not technically outside the stomach. It's into a cavity, into an opening. Those are exocrine glands as well, because believe it or not, the stuff in your stomach is not in your body. You've gone through your whole life thinking that, oh, I'm a two-legged, two-armed bag of substances, tissues, cells, everything else. But you're not. You're actually donut-shaped. The digestive system isn't part of your body. It's not in your body. It's a close association, but it's outside of your body. So, a brief recap on this section before we get into the integumentary system. These are questions that I might have asked via Turning Point, but I just want to make sure we're all on the same page. Cells are ordered in a specific manner and of specific types within a tissue. There are 4 main types of tissues that we looked at. You should be able to name them, hopefully. And how those cells are arranged, or what types of cells are there, dictate the functions of that tissue. Not only that, but how the cells are connected or anchored in place gives some tissues certain characteristics. Maybe it's waterproof or impermeable. There are a lot of tight junctions. We spent a lot of time talking about epithelium, the lining tissue on the surfaces of the body, into a lumen or on the skin surface. And connective tissue often supports that epithelium, provides structure. It's the scaffolding on top of which the epithelium sits. Do you remember what's between those 2 layers? The structure that's always found between connective tissue and epithelium? Yeah? Basement membrane, fantastic. Now everyone knows, so that if I ask that in Turning Point, you can answer it correctly. Basement membrane, absolutely. Okay, so that's our introduction to tissues. Let's focus on a tissue specifically, the integumentary system, which simply means the skin and its accessory structures. Largely means the skin. We're focusing on the skin, but the accessory structures like hair, nails, glands, etc. are included in the system as well. So let's briefly introduce this system. You will look at it in depth next week. We're going to look at the main characteristics, the main functions of the integumentary system. Well, first and foremost, the skin and its accessory structures are the bag that holds our internal components together. It's the barrier of protection between the internal and external environments. So protection is a really big focus in this section. The integumentary system is skin, glands, hair, nails. It helps the body maintain temperature. Maybe I should have warned you before I flipped to this slide. Who's been to Body World before? A couple people? So you've seen images like this in the background. These are plastinated cadavers that have been donated, and the artist in question will arrange the bodies in certain positions so you can focus on different systems or different musculature or the organs or how a body accomplishes a goal or a task such as childbirth, for instance. Really impactful exhibition if you ever get to see it. 2 years ago it was in Halifax, which is not helping you at all. 2 years ago it was in Halifax. I don't know if they plan on returning. When I went, I was in second year doing my anatomy undergrad, and I was the most critical person in the room. Like, you can't do that. That's not what muscle tissue looks like. Little did I know. Anyways, this is Body World. This is a real person. That's their skin. The integumentary system highlighted for all to see. Skin, hair, oil and sweat glands, nails, sensory receptors and organs, the associated blood supply. The skin is the barrier that protects us from the external environment, helps us thermoregulate, does some accessory functions like synthesizing vitamin D. And we'll look at later, it's also a site where you might be able to deliver therapeutic drugs through the skin. So a number of different functions, but let's focus first on the structure. When we say skin, what do we mean? What is the structure of the skin? The layers that we are concerned with are primarily two. The most important, I would say, is the epidermis, the uppermost or most superficial layer, the epidermis. We're going to look at that in detail. The dermis is just deep to the epidermis, and we're not going to look at that in as much detail. Epidermis is mostly epithelium, dermis mostly connective tissue, and we'll see that in detail coming up. You'll also notice on this slide, so you'll see epidermis is the uppermost layer on the right. All of this purpley pink area is dermis. And that purpley pink area holds most of the accessory structures. There's hair, there's muscles, arteries and veins, nerves, glands. We'll look at those coming up. But there's a lot of stuff in the dermis. Deep to those two layers is the hypodermis, which I really won't mention after this slide. It's mostly adipose tissue and some fat. There's some larger blood vessels. It's not technically a layer of the skin, but it does have some support role in anchoring the dermis and supporting the two upper skin structures. We will look primarily at the epidermis, followed by the dermis, and I'm not talking about the hypodermis anymore today. So let's start with that first layer. The structure of the skin. What is the skin made of? The epidermis is where a lot of the barrier functions come into play, the barrier and protective functions, and they're all due to these cells which comprise the epidermis. If you were to take the epidermis as one tissue and classify it, it would be keratinized, stratified, squamous epithelium. Keratinized, stratified, squamous epithelium. And what that implies is the cells contain a lot of this protein called keratin, which is really good for skin. It's structural. And stratified squamous epithelium, you know from the last few slides. By far, the biggest constituent cell in the epidermis are keratinocytes. You can see those on the top left of the inset figure on the right. They look really spiky. Those spikes are the keratin proteins. They're pointed in all different directions, and so there is support and rigidity in many different directions. It's fibrous. It protects against abrasion and wear. It helps the cells keep their integrity. 90% of the cells in the epidermis are keratinocytes. These are also the cells that make your skin waterproof. At a certain stage, which we'll see coming up, they secrete what are called lamellar granules. We don't really care about those other than to know that they make a hydrophobic coating. That is, a coating that repels water on the skin surface. So tough fibrous cells that make the skin waterproof and seal the barrier separating the internal and external environments. The next most abundant cell, at a measly 8%, are melanocytes, which are responsible for producing melanin, which are the pigments of the skin. Melanin granules dictate your skin color. There are many different types of melanin granules, and these are really interesting. They will when you expose them to the sun, when you're outside for a long time, melanin will actually move onto the outer superficial surface of the keratinocytes and absorb the UV radiation, just so the keratinocytes don't get damaged. They absorb the radiation, they change shape, and they change color. This is how you tan when you're outside for repeated hours on end frequently in the summer, which is the second week in July in Antigonish. In smaller amounts, we have macrophages, which are immune cells, and concern yourself with simply knowing that these fight off bacteria or foreign invaders. They're sprinkled throughout the skin so that if you get a cut and if bacteria gets in, these will hopefully deal with them so there's no infection. And there are a few tactile cells as well. Bottom right-hand corner on this figure, when you press your fingers against something, you deform, you change the shape of these cells. And they're sitting right next to a nerve. So as their shape changes, they touch and activate the nerve. And that nerve sends a signal to your brain saying, oh, I'm touching something right now. I can feel the pressure. I can interpret a tactile sense. So those are the 4 types of cells in the epidermis. And I'm going to jump to a slide showing you how they're arranged and then pull back really quick as I talk about the layers. So you can see the large majority of these cells are keratinocytes. Almost every cell on the slide is a keratinocyte. But you can see melanocytes sprinkled in here. There's one of those tactile cells, a macrophage. The rest are keratinocytes in various stages. This is the epidermis. Now, I'm going to pull back and then come back to this picture, but I've grayed out some of the items on the left-hand side for you, my right facing you. I want to introduce those layers first. The epidermis has 4 main types of cells. Keratinocytes are the most numerous type of cell. And they're arranged in 5 layers. I'll say 4 or 5 layers. From deep to superficial, they are the stratum basale, spinosum, granulosum, lucidum, which is the special case, and corneum. And I'm introducing them now. We'll talk about them in detail when I go back to that figure. Lucidum is here. It can be there or not. Depending on the type of skin that you're looking at, it might have the stratum lucidum. Consider for a minute the skin on the bottom of your feet. Really callous, hard, thick skin. Supports your weight on a daily basis. It's doing that for me right now. Endures a lot of stress, a lot of pressure. The stratum lucidum is there in that skin. It's a really hardened layer that helps support the skin. Lucidum is not in the thin skin on the back of your palm. 2 varying types of skin, very thick, heavy skin, contains the stratum lucidum. Thin skin does not. So let's take a quick look at the arrangement of these layers, how these cells are distributed through these layers, and what makes the layers different. The most deep layer is the stratum basale. It's a single layer of cuboidal cells that is at the bottom or the base of the epithelium. You can see these darkly staining cuboidal cells here. That's the stratum basale. And these are responsible for making all of the keratinocytes. All the other cells that are more superficial to these cells came from the stratum basale. It's full of stem cells that always make new keratinocytes. And so you can kind of think of this as bubbles rising to the top of your favorite pop or soda or other adult beverage. The stem cells in the bottom give rise to these keratinocytes that gradually migrate further and further out, more superficially. And so you're looking at the life stages of all of the keratinocytes as you move more superficially. Stratum basale has the stem cells. It contains the tactile cells. This is where you register press or push. And melanocytes here make melanin granules that filter out as well. Stratum spinosum is the next one. And I'm not going to tell you much more about it. This is where all the regular keratinocytes live. Those spiky keratinocytes that give the skin its shape and its structure and its qualities, they live in the stratum spinosum. But one thing that I will point out is the macrophage, that bacteria-killing cell, lives here. So if you have a moderately deep cut, the macrophages in the stratum spinosum can hopefully take care of that and destroy any foreign invaders. Where I think it starts to get interesting, though, is in the next layer, the stratum granulosum. And the name gives a sense of what's going on here. Granulosum. We've heard that word or a similar word already today. Granule? Granulosum? The keratinocytes in the stratum granulosum are dying. They're undergoing what's called apoptosis, a fancy scientific word for cells dying. Fascinating concept. This is actually a program. And your body induces this program when you get too old. Part of aging is thought to be due to apoptosis. But regardless, these cells don't get enough nutrients. Remember, epidermis is far from the blood supply. All of the oxygen and nutrients that get to the epidermis need to go from the blood supply deeper in the dermis, and then by diffusion make their way out. The keratinocytes in the stratum granulosum are too far away, so they die. But this is good because in the process of dying, they release lamellar granules, and now we start to create that hydrophobic layer, that waterproof layer. It gives this layer its name, and you can see them scattered around these dead, flattened keratinocytes in that layer as you move more superficially. Stratum lucidum, you can see there the whitened cells. They are a hardened layer that helps protect against impact and pressure. These are only found, like we talked about, in thick skin. It's 4 to 5 layers, and they're almost like stale or petrified keratinocytes. The dead keratinocytes that have hardened at the upper layers. What's interesting is you can recruit this layer. You can make this layer larger or smaller. If you start walking around without shoes and socks on, you'll notice that your feet become more calloused and rougher. It's this layer that's thickening in that process. If you work outside with your hands doing construction all summer, your hands will become more rugged and calloused. It's this layer that is thickening. And superficial to that, the footnote of the epidermis, the stratum corneum, which is 25 to 50 layers of dead skin cells that don't do anything and then slough off when they reach the top. That's what makes up dust in your house. That's what, when you use a loofah sponge or something in the bathroom, you scrub off all the dead skin cells. That's the stratum corneum. Dead keratinocytes that fall off periodically and are constantly replaced. So the life cycle of a keratinocyte starts at the bottom here and moves up. So that's the epidermis. You'll probably learn more in lab. That's what I want you to know for now, at least to prepare you for lab. The dermis, on the other hand, We're not going to spend a lot of time talking about it, except to acknowledge that it contains many interesting structures. This table is in your book mostly to acknowledge there's a difference in blood flow and a difference in the collagen and elastin fibers, but I'll talk about those on the next slide. This is largely connective tissue. Largely connective tissue. So there's some blood supply that gives substrates to the dermis. There's a lot of connective tissue that provides support and gives skin its shape and resilience and flexibility. It'd be pretty bad if my skin weren't flexible and I'm flailing my arms trying to describe things to you. But this is in the notes, the characteristics of the dermis. We can loosely break this up into papillary and reticular regions. I'm going to describe them on the next slide where I have a picture of those two regions. So, looking at a schematic of the skin, again we have the epidermis on top. This is a slightly more detailed picture, but it shows the same thing. Dermis in the middle that's currently highlighted in a light and a darker green, and then the subcutaneous layer at the bottom, which is mostly adipose or fatty. The two layers are the different green highlights. The papillary region, which has some blood supply and contains some nerve endings, and the reticular region, which is largely involved in keeping those major accessory structures. So the most superficial region is very loose and open. It's made of what's called areolar connective tissue, and you'll see this in lab. There are collagen and elastin fibers, and these are common fibers. When we talk about connective tissue, you'll often see collagen and elastin. Collagen are sort of the building blocks or the structural rigidity fibers. Elastin are the flexibility fibers. Really loose arrangement of these fibers. It's very airy. So there's abundant blood supply, and we have some other immune cells called phagocytes in this layer that, if you get a deep cut, can hopefully prevent against a bacterial infection. Macrophages in the epidermis, phagocytes in the dermis, at least in the papillary region. So loose, airy connective tissue. You can imagine what that would be like. Spongy but resilient, can move around. As you move more deep into the skin, less superficial, the reticular region is dense. Still collagen and elastin, but rather than this loose, airy structure, these fibers are packed together. And they point in every arrangement, again, to provide support and flexibility in many directions. This is highly innervated. There are a lot of nerve endings in the reticular region. And this is where we find many of the accessory structures that we're going to talk about next. Accessory structures like hair follicles, sweat glands, sebaceous oil glands. Most of the accessory structures are found in the reticular region of the dermis. So what are some accessory structures? I heard some sighs. Did you all get that? I'm going to give you maybe 10 or 15 more seconds, and then we'll move on. It's a great exercise in filtering out the extraneous material in words. You want to get to the point, definitive facts in your note-taking. Is your hand getting tired? Is that what that is? Yeah? So, accessory structures. What accessory structures are there? We're not going to look at all of them. I want to show you some that are somewhat interesting. One of the most obvious is shown here. Along with skin, you'll often find hair. It's present on most body surfaces, except the palms of the hands, the soles of the feet. and some other surfaces as well. What does it do? Is it just an evolutionary remnant? What does it do? I should mention, since this is going on YouTube, this is copyright Game of Thrones, this background picture. I don't make any claims to having made this, etc., etc. Don't sue me. Why I chose this picture will be obvious in a second. So hair itself is composed of dead keratinocytes. We just spent a lot of time talking about those in the epidermis. And on the next slide, you'll see how there's these really tightly packed cylinders of keratinocytes, which is hair. These dead keratinized epidermal cells are packaged together to make hair. The amount of hair, the color of hair, the type of hair, straight, curly, fine, depends largely on hormones. Genetic influences and hormones during puberty really affect the thickness and distribution of hair. So beard hair, for instance, that starts to be developed in males during puberty is subject to all of the raging hormonal influences that occur at that time and can have a different shape and structure and color than the rest of a person's body hair. Hence the picture in the back, Dorman Giantsbane having a clearly fiery red beard and blonde hair. Hormonal influences. Color of hair also affected by melanin, type and amount of melanin. Just like the skin, hair is made of keratinocytes, blonde hair, brown hair, red hair, affected by the type of melanin. So you can see those fused dead keratinocytes in this electron micrograph in the middle. This is the hair shaft, and all of these discs that look like little ridges are compressed dead keratinocytes. So we have the hair shaft above the skin. That's the part you can see. Just below the skin is the follicle, and then deep within that reticular layer of the dermis is the root. I'm not going to spend a lot of time talking about the different types of hair, but you can see the bulb and the projection on this cartoon figure over on the right-hand side. So the shaft at the top, the follicle below the skin, and the root is this bulbous shape inside the dermis. While we have this picture up, we can also see some other really interesting structures. So hair is one accessory structure, but you might have noticed if you ever get a tingle or feel cold or are surprised or shocked, your hair stands on end and you get goosebumps. That's the action of these muscles, these very tiny muscles that attach to the hair bulb. It's called the erector pili muscle. It makes your hair stand up. And maybe it serves little purpose now, but at one point, hair standing up made a larger, gave it a better ability to trap air. So made the insulating layer around your skin bigger. Your hair stands on end. There's a greater ability to trap air, not hair, in that region, which can help warm you in situations where you're cold. And you can obviously understand the more hair you have, the more that trapping ability, and the warmer you can stay. Also on this figure, a pretty obvious accessory structure is this squiggly and also long gland. That's a sweat gland. You might not have ever seen one before, but you know how it works. And this is also the site, remember, high nervous innervation in the dermis. This is a plexus of nerves, or a network of nerves that wraps around the hair bulb and can tell you, without you even looking to notice, oh, there's a bug crawling on me, or someone's going to grab me or try to tickle me or something. Your hair moving, unbeknownst to you, is registered through this hair plexus that gets sent to the brain to be interpreted. Other accessory structures in the skin. Glands. One that we haven't acknowledged yet or talked about is a sebaceous or oil gland. If you're traveling on an airplane for a long time and then you get off the airplane and you have this gross, slick, sticky layer on top of you, or you haven't showered for a couple days and you just feel kind of grimy, that's the action of these sebaceous oil glands. These happen to be connected to hair follicles. This little appendage on the way out of the epidermis is the sebaceous oil gland, and it lubricates the hair as it leaves the body. It continues to excrete oil these are collected in crevices, areas of the body where there tend to be a lot of friction and rubbing. And the oil glands help to lubricate those areas. So you'll find often a lot of hair and a lot of oil glands in places like armpits, for instance. There's a lot of moving back and forth, and these help to reduce the friction in those crevices. Type of glands, sweat glands. An exocrine gland that delivers sweat onto the outside of the surface. The eccrine sweat gland is different from one other kind that I'll tell you in a minute. The eccrine sweat gland is what you think of when you think sweat gland. Salty fluid. Lots of fluid as you play basketball or go for a bike ride. That's what I'm showing you over here on the right-hand side. This is an eccrine sweat gland. I'm not showing you its counterpart, the apocrine sweat gland, which you can think of as a combination of the first 2 points. It's a sweat gland that secretes an oily substance, not salty fluid, but an oily substance. And again, these are often found in those crevices to reduce friction. Since we're on this slide, and because you probably are familiar with these, ceruminous glands are a very specialized type of sweat gland found only in the ears that make earwax. Unfortunately, they're always active. They're modified sweat glands that produce a rather thick, oily, not even fluid, you could say, in the ear canal. So a few different types of glands. My favorite is a sweat gland. That's what I've studied, not the gland specifically, but the sweating response. That's what I've studied for a lot of my PhD and what I do research on, which you'll actually find out more about in Dr. Rasmussen's course as we come in and do guest lectures. You'll see that in November. Sweat glands, very cool, help keep the body cool, and that's all I'll say about that right now. So let me just check, can I do an abbreviated 5-minute synopsis of functions? I think so. I want to get through this because we want to start with the nervous system tomorrow in order to stay on pace and on track. We've covered a lot today. I get it. There's a lot of information. You've written a lot. There's some overload. I'm going to limit our discussion of functions of the skin to 2 or 3 main areas that may or may not be applicable. The first, thermoregulation. This is a list that you can write down. I'm going to focus on thermoregulation 1, and then the last 2, absorption and synthesis of vitamin D. But we've already talked about how the skin helps to protect our internal organs from the external environment. We've talked about sensations. The other functions of the skin are in thermoregulation. So a sweat gland delivering that salty fluid onto the surface of the skin, as seen here, allows that fluid to evaporate. Evaporation is an endothermic process. It requires energy. It gets that energy from the body. You lose heat through evaporation of sweat. We can only do this because of sweat glands distributed everywhere in the body. And maybe you're like me. I just noticed that my hands are really sweaty now too. It's like when someone talks about posture and you're like, oh yeah, I haven't been sitting up straight for the past few hours. You guys have nice chairs though, so relax, feel free. So the skin is the primary site of heat loss in the body. That's why we're such good hunters. That's why we evolved better than lizards did, or dogs. They don't have sweat glands. Blood flow to the skin brings the hot stuff that needs to be cooled, and then the sweat that evaporates cools the blood. Great site of thermoregulation. In the process of sweating, you get rid of some substances. First and foremost, you get rid of sodium, some other ions. Urea is found in the sweat in small amounts. You can excrete some substances, but it's not really a major function of the skin. What is becoming, I don't know why there's a space there, a potential therapeutic area is in the absorption of substances through the skin, the absorption of drugs through the skin. Anything that is really small, a steroid derivative, non-polar, so it's inert, can be administered and travel through the skin. And this is the schematic for a company called Delivra. They have some research going on with my alma mater, Guelph, and UPEI to look at transdermal administration of creatine to see if that's a way that it could be supplemented. Delivra also is producing a method of cannabis delivery in our new regulated landscape, again to be delivered through the skin, but the point is this vehicle might be able to provide a number of different therapeutic strategies to the body, put it that way. I'm not going to say the last one. Vitamin D, you know that your skin synthesizes vitamin D. It's not really the focus of this course that vitamin D gets synthesized. You'll talk about that in nutrition. Let's just assume that when you are out in the sun, which is never around here, vitamin D is synthesized into its active form. So we're going to come back. We'll do a couple recap and summary questions on Turning Point tomorrow on this material. Then we will start with the nervous system, central and peripheral nervous systems. Have a wonderful Thursday.